Now we are going to talk about how arabinoxylans are changing during the mixing, fermentation, and the baking process. Again, we have series of studies on arabinoxylans in refrigerated dough, but this is another example of why arabinoxylans are important and what happens to them during the mixing process and the fermentation process and the baking stage. And here we use three most commonly grown spring wheat varieties, but they have different genetic background. And then we collected those samples at different mixing and fermentation stage and also baking stage. But this table shows proximate analysis of the wheat varieties. So they had similar protein content and ash content and also very similar moisture content. And here we looked at, again, we had three different varieties, and each bar with different color is showing a different wheat variety. We were checking the changes on the arabinoxylans solubility during the bread making process. So in the flour, the amounts are lower, so it's between 0.2 to 0.3%. But as we move into the fermentation process, after mixing and the punching and the molding, again, is more progressing in the fermentation process, we see that soluble arabinoxylans are increasing. Of course, if something is increasing, we need source. So what happens is, due to some enzymatic reactions in the flour, the water insoluble arabinoxylans were converted to water extractable ones, because we had the completely opposite trend for the amount of water unextractable arabinoxylans here. And then there were also some changes in terms of very fine details of arabinoxylans during the mixing and the fermentation process. And here we used some cartoon version. Remember, arabinoxylan structure, we can have no substitution, or we can have one or two substitutions, and they will have different properties. And using very advanced chemistry technologies, we can actually quantify and measure how much of the xylose has one or two type of linkage using proton NMR. So we actually studied again what happens to arabinoxylan, and we proposed a model that first the water unextractable arabinoxylans, again this is the backbone is xylose, and then we have substitution of arabinose. And the water extractable arabinoxylans at the beginning, like they have so much arabinose, they are very substitute. As the fermentation progresses, so we also have removal of changes of arabinoxylans in the water extractable arabinoxylans. The xylose residues are leaving. But then very highly substitute portion of unextractable arabinoxylan is released because of enzymatic reactions, and they are moving into the water extractable portion. And then we were able to actually control these changes and monitor these changes using, again, advanced chemistry technologies. So this is just one example how we can study arabinoxylans, but then there are some changes during the mixing process and fermentation process. Again, they are very important for water absorption capacity. Another important non-starch polysaccharide is not the relative abundance, the amount is not as high as arabinoxylans, but the other one is beta-glucan. Again, we hear beta-glucan a lot for oat and barley. So beta-glucan is actually very similar to cellulose. So what cellulose is, the chemistry of nature is very interesting. I find this very fascinating, because if you look at this starch molecule is made of glucose, and the glucose are linked to alpha linkage, and I just use this shape of downward linkage. So this is glucose, this is another glucose, and they have the alpha linkage. This is how starch molecule is linked to each other. With cellulose, which is water insoluble and is a very rigid molecule, it's also glucose, but the glucose is linked to upward fashion. This is beta linkage. And the alpha versus beta, this is the same glucose, but how they come together in three-dimensional system makes it a completely different molecule. It's very interesting. And starch, it's very essential for us. We need to consume with our diet and convert to glucose. But then cellulose, it's very rigid. This is why your tables, your doors, your wood door is going to be very firm, is because of cellulose. So cellulose is all the trees, they have cellulose, and they are not water soluble. That's why trees do not melt when it rains, right? So beta-glucan is very similar to cellulose. It's beta-1,4-linked glucose, but there is one small difference. Here, again, this glucose beta-1,4 linked I just described, but every 3 or 4 glucose molecule, we have the 1,3 linkage. 1,3 means that number, the carbon number in the glucose I have shown you during the non-starch polysaccharide discussion. Then this molecule becomes soluble, and then there is great amount of biological functionality. So it's the size is big, but still is in water soluble. Is why? Because of that one deviation in the structure compared to cellulose. And it is considered as dietary fiber. You all know that what dietary fiber is. Dietary fiber is mostly plant-based compounds we take with our diet, and they are converted into the helpful compounds in the digestive tract, but we cannot convert them to glucose. They do not contribute the calories, but they have additional health benefits for digestive tract. We are going to talk about dietary fiber in the following lectures. So beta-glucan has functionality for viscosity, and also it's an excellent dietary fiber. And we already talked about the structure. So beta-glucan, compared to wheat and rye, there is higher amount of beta-glucan in oats and barley. It's the major. And then actually the beta-glucan also could be found in the endosperm of these cereals, additional to bran portion. In bakery products, they have high water retention capacity, just like any other non-starch polysaccharide. And they also have positive impact on the stabilization of gas cells. And for beta-glucan, we also can find them in different sizes. So if a molecule has smaller size, typically it has less viscosity. If we have higher molecule size, then there is higher viscosity. And if the molecular is too big, then it's going to be water insoluble. It's just going to precipitate. How much viscosity we are going to have, how much solubility we are going to detect for beta-glucan is going to be controlled by the size of the molecule. And then also how much of that 3 linkage we have. And the processing conditions will have very profound effect on the solubility of the beta-glucan. And there is also some gel formation with beta-glucan. So you will hear gel for starch. We will also, you might hear oxidative gelation for wheat. And gel is, in general, definition of gel is it's a 3-dimensional network that is water is entrapped into that system. Beta-glucan also could be part of a gel system. And with that, we are going to stop here. In the next lecture, we are going to talk about starch.